Hello, everyone. This is your recording for the first week of April. So I'm starting us out with our homepage, checking out what we've got going on for this week. So this week you have 3 workbooks, and then we're going to be doing a lab this week. I'm going to talk a little bit about it at the end of this recording, and then you'll have a separate recording to look at specifically just for that assignment. 2 of the workbooks are in CK12. Quick note, I had a couple messages from people who were having trouble accessing the CK12 assignments, and I did have some insight. It seems like one of the issues that happens is if you don't have your cookies enabled. So I do have a link in both of these assignments showing you how to make sure your cookies are enabled in case you are having issues accessing those CK12 assignments. All right, and then we'll have optional office hours Monday and Friday of this week. Keep in mind these office hours are optional unless you currently have a D or an F for Q4. If you're in that boat, then those are required office hours so that we can get together and get you back on track. If you have an A, B, or C this quarter, then you do not have to come to those office hours, but you are more than welcome to if you need help with anything or have questions. Announcements for this week, I will be out for testing Tuesday and Wednesday, so I may be a little bit slower responding to messages. And then the other thing to remember is that you don't have a quiz this week, but this lab that you've got, you're going to spend 2 days on it, and it's going to count as your quiz grade for this week. All right, so if you go into your acids and bases slides, remember these are going to be posted into your resources as always. I do have reminders about this week's assignments right at the top of these slides. I'm also linking the recording that you're watching now at the beginning and then the lab recording in here as well. So today we're on Unit 8, Lesson 9, talking about acids and bases. We're going to start by thinking about what are acids and bases. Then we're going to talk about their properties a little bit. We'll talk about what pH means. We'll talk a lot more about that next week, but we'll start talking about it a bit this week. We'll talk about what pH indicators are, and then the demonstration that I'm going to do is actually going to be part of that lab altogether. So that'll actually be in that separate video that you get. Some vocab to keep in mind this lesson. So lesson 8, we learned all about solvent, solute solutions, and aqueous solutions. When we're thinking about acids and bases, we think about the acid or base as the solute, and typically water is our solvent, and that would make these acids and bases aqueous solutions. Some new vocab that we've got for this week is acid, base, the word alkaline, pH, and indicator. So keep an eye out for those terms as we go through. All right, so for this week, you don't actually have to know what's on this slide. It will come back up again next week, and it'll be much more important for us to understand next week. But I just wanted to give you a preview of it now so that when I'm talking about acids and bases, you have some context for what these terms actually mean. And then next week, we'll talk more about how we figure out what we actually mean when we say things like hydrogen ion concentration. But for now, I just wanted to give you these kind of simple definitions. So acids are any substance that when we dissolve them in water result in an increase in hydrogen ion concentration. So those are my H pluses. And bases are any substance that when I dissolve them in water result in an increase in hydroxide ion. That's the negative OH concentration. There are more precise ways that we can define acids and bases, but for our class, these definitions are going to be fine. We don't need to get too much into the nitty-gritty of these definitions. There are more, but this will work for us. And then the word alkaline is just another word for basic. Those are synonyms. So if you see this is a basic substance, that's the same as saying this is an alkaline substance. It's not the same as the word base. You wouldn't say this is an alkaline. You would say this is alkaline. It's used to describe, like the word basic is used to describe, so it's an adjective. When we're talking about the properties of acids, they typically taste sour. So some examples are lemons, tomatoes, citrus, vinegar, and soda. Soda is actually very acidic. That being said, never go around tasting non-food items to determine if they're acidic. You don't want to go around tasting random things, random solutions you find and saying, is this acidic? Let me taste it. We don't want to do that. That's not safe. But if it's a food item and you want to know if it's acidic or not, whether or not it's sour is a good indicator. Acids will react with metals. This is why you actually can't cook acidic foods in a copper pot or aluminum pot. I had a friend once who put orange juice in her aluminum water bottle and then said like her teeth hurt for the rest of the day because it just creates this really, really horrible combination because those acids are actually breaking down the metals that they're coming in contact with. So soda cans and other, like I have a LaCroix in front of me right now, that can, any aluminum can that has a soda in it is going to have a thin layer of plastic inside it that prevents the acid in the soda from corroding the can. So like I said, acids are corrosive to metal, but they're also corrosive to skin. And particularly when we're working with stronger acids, those can be really corrosive. Corrosive just means that they're going to react in a way that's harmful. So the acid in the soda is corrosive to metal, meaning it'll break down that metal over time. This is also how stomach acid works in our stomach. It corrodes and breaks down our food through that acidity. And later we're going to talk about something called litmus paper. And litmus paper is an indicator, and if it turns red, that tells us that we have an acid. Properties of bases. They taste bitter instead of acidic. They are somewhat common in foods, but not quite as common as acids, I would say. They're more common in things like cleaning products, so we will see them around, but not as often in food. So some examples are baking soda, Tums, and soap. And again, never taste non-food items to check for acidity. Bases feel like slippery to the touch. So for example, when you put soap on your hands and you have that slippery feeling, what's actually happening is that bases, when they come into contact with your cells, react with the cell membranes and break open your cell membranes. So what you're actually feeling isn't the base itself. It's not even the soap. It's the actual, the inside of your cells that you're feeling in your hands. So you're actually feeling your own exploded cells when you touch soap. They do not react with metals in the same way that acids do, but they are still corrosive to tissues. And when I say tissue, I mean like skin tissue. So this is why we always want to wear gloves when we handle any strong cleaning products. You never want to touch your hand directly to bleach or anything like that. And when we put litmus paper in contact with a base, it will turn blue. All right, so moving into pH. pH is something that we use to measure how acidic or basic something is. Next week, we'll learn how to calculate the pH from a solution. But for now, we're just going to learn what it means. So pH, notice that it's pronounced pH, just letter P, letter H. And when we write it with a lowercase p and a capital H. It's a measure of how acidic or something or basic something is on a scale from this should actually say 0 to 14. Fixing that. From 0 to 14. A pH of 7 is neutral, so right smack dab in the middle is neutral. Pure water has a pH of 7 and is neutral, but you likely aren't coming into contact with pure water. You're probably coming in contact with tap water or bottled water, which is going to be anywhere from 6.5 to 8.5. So either a little bit acidic or a little bit basic. And then there's also something like bottles like Smart Water or other brands that advertise being pH balanced. What they basically those are just water that's a little bit basic or a little bit alkaline, we could say. So they usually have a pH of around 9.5, and we'll look more at smart water as one of our examples in our lab that we do. So anything that has a pH that is less than 7 is acidic, and anything that has a pH that is more than 7 is alkaline or basic. We can remember this by thinking alphabetically. So below 7 comes first, that's acidic, A. After 7, that's lower in the alphabet, that's a B for basic. Anything that is lower in pH means it's more acidic, and higher pH means it's more basic. So a pH of 1 is very, very acidic. A pH of 3 is acidic, but not as acidic as something with a pH of 1. A pH of 6, only a little bit acidic. pH 7, neutral. pH 8, only a little bit basic. And then pH 11 would be very, very basic. All right, so here's our scale. We've got some common items in here as well. So 0, the most acidic thing we can think of is something like battery acid. And then all the way on the other end, the most basic thing we can think of is something like a drain cleaner is very, very basic. The colors that we see on this scale are also, they correlate to the color that this substance would turn a universal indicator paper. pH indicators are made from chemicals that change color in the presence of acids and bases. They're usually used with a small dropper, but they're used in really small amounts, so they don't end up changing the overall reactants and products of the reaction, but they do change color based on the reaction, and so we can use them to determine what the pH of a substance is, or when we have a reaction, what the pH is at the start and the end of a reaction. Some pH indicators have a really limited color range, and they tell us about if we're looking at something that has a range of only 1 to 5, it's going to tell us really, really specifically within that what that pH might be. Whereas other indicators, like the universal indicator, have a wider range that we can use to look at. And so they may not be quite as precise, but they give us information when we may not know as much going in. And the only one that you're going to need to know the name of is litmus paper and maybe universal indicator, but really I want you to know what litmus paper is, what it looks like. The other ones, I'm going to tell you a couple names, but I'm not going to ask you to tell me what they are or name any other indicators for me. So there are two types that come in this little paper form, and one is litmus paper. Litmus paper comes in either red or blue. And usually when testing a substance, we'll take a red litmus paper and a blue litmus paper and put them both into solution. And if the blue one turns red, then we have an acid. If the red one turns blue, then we have a base. If the substance is neutral, they're not going to change color. If I put a red litmus paper into acid, it's not going to change color. If I put blue litmus paper into base, it's not going to change color. So the only change we'll see is going from blue to red or going from red to blue. So remember, acids turn red and bases turn blue. The other and often more useful type of indicator, so the thing about litmus paper is it can really only tell me either acid or base. It can't tell me how strong the acid is. It can only tell me if it's acidic, basic, or neutral. So it can sometimes be very useful, but other times not quite as useful if I want to know, is this a stronger acid than something else? And if I want to know a little bit more information. So in that kind of situation, something like a universal indicator might be a little bit more useful, because in that case, when I dip this paper into solution, it's going to change color according to this scale. So it'll turn red if it's really acidic, and then if it's only a little bit acidic, it might turn a little bit green, and then if it's really basic, it'll turn blue or purple. So it gives us a little more info. So I want to make sure that we would know how to read these. So let's look at some litmus paper. So this is a situation where we have a student, they've dipped both red and blue litmus paper, just like they're supposed to, into 3 different solutions. And we know one is an acid, one is water, and one is a base. So what I want you to do is pause the video for a second and see if you can figure out which one is the acid, which solution is the base, and which solution is neutral, just looking at how the litmus paper has changed. So go ahead and pause and think about that for a moment. All right, hopefully you paused and thought about which solution was acidic, basic, and neutral. So the one on the left is acidic. I know this because if I look at the very tops where the paper is still dry, I can see which paper I started with. So I started with, if I'm looking at this left solution, I started with a blue litmus paper on the left and red litmus paper on the right. And I can see that when it was dipped into the solution, the one on the left that was blue has now turned red, and the one on the right was already red and then stayed red. It didn't change color. So that, because blue turned red, that's acidic. My middle solution is neutral. Neither paper changed color, so that's got a pH of 7. And then my solution on the right is basic. Again, if I look at the top of the paper where it's dry, I can see what they started out as. I can see my blue litmus paper stayed blue and my red litmus paper turned blue, so that's a basic solution. B for blue, B for basic. All right, what about using universal indicators? So now we have another student. They've tested 4 substances using universal indicator paper. And I'd like you to cross-reference with the chart that's included in my indicator paper and try and determine what do you estimate the pH for each of these solutions to be. You'll notice some of these colors are very, very similar. So you might have a little bit of a range. If I look at like 10 and 11, they're like the exact same color. So you could just say 10 to 11. Or if you're looking at maybe 3 and 4 look the same color, 8 and 9 look the same color. So you can put a little bit of an estimate if you need to. It doesn't have to be the exact pH. Go ahead and pause here and try and figure out these pHs. All right, here is the estimate I came up with looking at this indicator. I see the first one is pretty purple, looks about 12 to 13 to me. The second one is this really pretty teal color. I'd call that pH 10 to 11. It looks like that color. Middle one is a nice light green. That's our neutral. That is a pH of 7. The next one that looks pretty yellow-orange to me, so that's pH of 6. And then last, that one's pretty darn red. You might have called that a 3 to 4. I think it's a little bit closer to the 2. Looking at it now, I actually think it might be closer to 3, 4, but when I looked at it before, it looked more like a 2, so that's what we called it. But again, we're doing a little bit of estimation here because it's not super, super exact. If you're actually in a lab and you're doing something with acids and bases, you'll see people take their little litmus paper and then they'll go up to the chart and hold it up and kind of compare. They'll move it along the chart and you'll see them trying to line up which color matches the best. I can't do that on a computer, so I was kind of just trying to eyeball it. All right, so some other indicators that exist that we won't be using for our class, but just to know that they exist and what they look like. Again, you don't have to remember the names of these, but I just want you to see some other ones. So some of the big ones are bromethyl blue, and it ranges from a yellowy color to a really royal blue color for bases. Methyl orange is more similar to litmus paper in that it really only has an acidic and basic color. It doesn't really have a range in the way that bromethyl blue does. And same with phenolphthalein. Phenolphthalein is often used for something called titrations. And so what they'll do is you'll drop base into your acid, and then as soon as it switches from colorless to pink, we know that it's gone from acidic to basic. And so that's one of the uses that we can use for indicators. We aren't going to be doing titrations in this class, but just wanted to expose you to it. And then my favorite is red cabbage, which is a natural pH indicator. And so this is what I will be using in your lab for this week. So what we're going to do is I am going to boil up some red cabbage, and it's going to work as a natural pH indicator. You are welcome to try this at home. You put the red cabbage, you boil it up, and then you take the water that comes off. And if you add that to various solutions, it will change color based on the acidity of that solution. So that's what I'm going to be doing for you guys. So here's what your lab is going to look like. And remember, this is your quiz for this week. So part 1, you've got a couple of pre-lab questions. You can already do this part now after having gone through this video. So first one, what are some properties of acids? What are some properties of bases? And then what is the purpose of an indicator? Part 2, you're making a prediction. So I tell you what substances we're working with, and I want you to just make a prediction based on their properties. Do you think that these will be acidic or basic? Please, what I don't want you to do is go in and look up the pHs. You'll spoil all of the fun if you do that. Your predictions don't have to be correct. This is just your guess. I fully expect that some of your predictions will be incorrect, and that's fine. That's part of the point of doing a lab, is so that we can adjust what we thought was going to happen. And then I'd also like you to look through these substances and decide what you think the strongest acid will be, and then what you think the strongest base will be. Then, so this part you'll need the lab video for. So then I'll actually have all of my different cups laid out, and I'm going to add indicator into each one. You're going to color in the different cups to make your observations. So for example, for that first cup, you're going to write in vinegar, and I'm going to have a cup of vinegar in front of me. And then when I add the indicator, it's going to change color. So what I'd like you to color in on that cup is the color that it turns after the indicator is added. And then finally, part 4 is your analysis and conclusion. So after you've gone through and filled in colored all of your cups, you're going to fill in this data table. So you're going to write in the color that was observed. And then based on that color, you're going to use this chart up here to determine what pH you think it is. So let's say we put, I put my indicator in one of my solutions and it turns lime green. Then for the estimated pH for that substance, I'm going to say that's about 12. Again, you'll have a little bit of estimation, and that's okay. And then finally, based on the pH, you'll decide, is it acidic or basic? So if I had one that was 12, I would know that that was basic. So I would write basic in that last column. And then making your conclusions, you're just going to decide what's the strongest acid, what's the strongest base. So whatever has the lowest pH will be your strongest acid, and whatever has the highest pH will be your strongest base. And then you're just going to tell me, does that agree with what you predicted or not? All right, so that's everything you guys have coming up this week. If you have writing this week, have good luck with your testing. And then if you would like to come to office hours, I am more than happy to see you Monday or Friday if you have any questions. All right, thanks everyone. Bye-bye.